Okay. Hello. I actually can't see my own screen. So let's do that. Hello, students. Can you hear me? I might be a little bit distracted right now. There are things that you don't really want to know. Hello, is there life? Is there life out there? Hello, life. Hello, human being. So when does the you're here for the room clean? You know, actually, I made it worse. So my lab, before I left yesterday, my lab told me there's a problem with the MRI button box. And so, like, this is stuff I'm doing right now on my table. I took apart the box and I'm trying to figure out what's wrong with it because, well, I know what's wrong with it. I don't know really how to fix it. Well, I do know how to fix it. It's just not very elegant. So what happens is, like, this is totally this is remotely not even relevant, actually. So when, like, the subjects press a button right now, the button gets stuck on its way back up, and so we're having some of these issues right now. So it's not a problem with the button itself. It's a problem with these stupid holes. And this whole thing cost, like, maybe, let me think, it was like $4,000, $5,000 US dollars for this piece of piece of crap. So one solution is to make the holes bigger, but that's gonna create a lot of dust in my office. The other solution, which I came up with, is we're gonna just raise the buttons so they're so high that they don't even go underneath the shell. Just not so elegant, but it works, given they need to use it tomorrow. I'll start room cleaning if, like, you know, if there are really no questions, if that's really what you want to watch. But, like, there's a lot to clean. There's a lot of people texting me too. Did you guys start studying? You guys just here to watch my room cleaning? That's interesting. We're gonna get more class attendance if all I do is stream room cleaning. We're really just here for room cleaning? Oh, that's so adorable. Have you guys all seen my office? There's actually a lot of junk in my office. There's a lot of cup noodles in my office, actually. Not eaten. There are questions, right? There are questions. It's not just about my office. I do realize there will be like a 10-second delay. So I'm going to put the music back on. Oh, hey, we have a question. We can also put the music back on. It's not a big deal. Hold on. Let me know if it's too loud. Okay, so the question is, growth cones. Are they on the dendritic side or axonal side of growing axons? Jade says both. Both. I actually did write your exam yesterday. I feel quite good about it. You guys might not feel so good about it. I'm kidding. Yeah. There are a few surprises in there. Yeah, Jade. Yeah, so that was an easy answer. Both sides. This thing is really annoying. Oh my god, this is so ridiculous. Look, it's literally, I don't know if you can see it, like the buttons are so high. Don't even know why we have a top cover now. It doesn't get stuck. What is the chapter that you're most worried about? Maybe we can do something about that. Could you please re-explain the side for a visual about lesions to the radiations of view 1? Yeah, sure. I mean, I also had, can you tell my massive distraction? Okay, let me just put these screws away. Okay, I'll go in chronological order. So let's do the vision lesions first. So was that week? That was week 8, right? All right. This one, right? Yeah, this one. So here, I think I said this in my, so we made a little video for Mr. Samuel, and that's actually, I left it still up, so it might be worth also revisiting that. But this is the sort of diagram you want to remember. Yep, great. I see questions coming in now. Okay, maybe we really do mute this thing now, or really turn it down. I think it maybe it's actually distracting me more than anybody else. Okay, so the thing to remember about this is that we've got basically 2 halves to each nerve that exits the retina, and each half is sort of responsible for a different segment of the visual field. And so the reason why I say that this is kind of the image to study is because if you can kind of reproduce this, on the exam, if a question comes up, then you can kind of just say, you can just follow the path, right? If we lesion here, then what was that blue line representing here, right? And so you can kind of, then you don't really, it just doesn't need to be brute memory in that sense, if you can understand how the fibers are working here. So very quickly, the first kind of trick is to remember that actually both nasal and temporal fibers encode part of the kind of central visual field. The only difference is that the nasal side of each eye is the only side, like it's the only half fiber that will carry the ipsilateral peripheral field. So you can kind of see that wedge over here. So for that reason, if there's any lesion that involves this nasal chunk of each eye, then that will always result in a complete loss of the ipsilateral periphery. Okay, and so yeah, I don't know. I don't know if that helps. So that's the nasal. But remember, of course, the nasal also encodes part of the central field. So you see that whole wedge over here. But the question is, if I lesion here, why is it that I just have a peripheral loss? Well, also remember that, of course, on the same eye, I've got a temporal half that will also cover much of my central vision as well. But I also have the contralateral eye, right? The contralateral eye over here, it's temporal fiber that is also giving us data from the central part of the visual field. So you've got a lot of overlapping representations, but the only thing that is not duplicately represented is the nasal half there. I'm not sure how much we need to review here, but I think from a study point of view, this is the stuff to get straight here. So we've got the temporal retina doing the central field of view, but also representing parts of the contralateral visual field, and then the nasal fiber representing parts of the central visual field, but also our ipsilateral peripheral vision. So then you can kind of cross over, of course, keeping in mind that if there's a lesion down here, right, then you've got to kind of draw it out. Hey, we're past the chiasm. So something else is happening there. So these three here, right? These three here would be the things just to, after you study that, these three are the things to kind of refresh your memory on, right? So here's an example where we've transected both the nasal and the temporal half of the left eye, but remember that it's the left temporal, the left nasal retina that is taking care of this side, so that's why it's all gone. So yeah, and so why when we lesion here over here the optic tract, do we now have a complete loss of this right hemifield? Well, it's because now we are past the chiasm. Yeah, is that okay? Oh, but I think you were asking okay, sorry, I missed that. You were asking specifically to the radiations or V1. So somehow we could have that could have been a lot quicker. Sorry, I didn't totally read that. Yeah, so that's this one over here. Still sort of thinking about my button box. Yeah, so remember that the radiations as we're leaving now the thalamus to V1. Remember, actually, did I write this down? I didn't, because there was the Bohm's loop, right? Well, I won't ask you for the name, so it doesn't matter. But remember, there's an inferior chunk where it dips down towards the temporal lobe and then coming back up dorsally before it goes to V1. So we've got inferior radiations coming down through the temporal side, and then the superior radiations before hitting V1. And so it just depends on whether we are lesioning the inferior or superior chunk. If we're lesioning now the optic radiations at the kind of the bottom part, the inferior part, then we lose actually the superior quadrant. And if we lesion the superior radiation, then we're going to lose the inferior quadrant. So that's the main thing. That's the main thing to remember there. By V1, we have, well, it depends on where in V1. But if you lose the entire V1 of, let's say, the left side, then you're also going to have a complete right hemifield loss on the contralateral side. Is it C-ray? Does that help? I did too much, apparently. I didn't realize you were only asking about the radiations. Yeah, so here the only thing to remember is what happens if it's just inferior, what happens if it's just superior, and then what happens if it's the whole V1. And, you know, I have to tell you that it's very rare clinically to see a whole V1 lesion. Where it's super tricky is like it's a chunk of V1, so it's not a complete loss, but only a small chunk. Arcelia, that's a really cool name. Didn't know we had an Arcelia in our class. Is it because the grade sheet has your Chinese name? Can you explain the phase locking thing? Okay. Phase locking, so that's week 9. You see, there was a reason why I set this for 1 o'clock. I bet you guys were all sleeping until now, huh? See my very poor impressions of you guys? Phase locking. I guess it's here. Right, the slide, Arcelia. Yeah, so the confusing part is that, you know, prior to this slide, the story was about how frequency is represented by the maximum point of the traveling wave. I see. I don't know where Dorita came. Honestly, I have a rougher time with my name. My parents said it came out of a name book, so don't know how to respond to that. There wasn't too much thought into that. So I think the confusing part about this is we know that already we should be able to extract frequency data by looking at the maximum point of the vibration on the basilar membrane. But of course, at some point, that also needs to be represented in cortex, right? That same data needs to be encoded in cortex. And so the question is, how do we do it? So tonotopy, of course, is the easiest one, because that follows by, hey, wherever on the basilar membrane equals to whichever ganglion cell, which equals to whatever cell that, is our whichever frequency location on A1, right? So if you remember that slide with the, do I have that slide here with the frequency representation in A1? We had that somewhere, right? Where's the first slide with a brain on this lecture? Yeah, here, right? So this concept's pretty easy, because that's sort of how we thought, frequency should just be straightforwardly encoded. We've got which segment on the basilar membrane has the maximum vibration, therefore where on the spiral ganglion, all the way up through the superior olives and all the way up to A1. Then we have a corresponding tonotopic representation. But it turns out that there's another way where we can resolve frequency information. Holy cow, where is that slide now? Gosh, phase locking, where are you? Here we go. So it turns out that at very low frequencies, so we're looking at very, very slow traveling waves. What we actually find is that firing is actually locked exactly to like every cycle of the stimulus. So here's the actual stimulus wave, and here's, let's say, the response of an auditory neuron, and you see that it always spikes like at this particular phase of the actual physical sound stimulus. So we find that in addition to tonotopy representations, we actually find that the auditory system uses phase locking as a way to retrieve the actual physical stimulus that has occurred. So in fact, this is what I wrote here. At very low frequencies, it turns out that phase locking alone is already sufficient to give us this information. And then the trouble is at high frequencies, if you can imagine a situation where, so you can imagine just there's a physical limit. We talked about the action potential, and we talked about the absolute and the refractory periods. So there's a physical limit to how often our neuron will be able to fire. So phase locking is completely not feasible at very high stimulus frequencies, right? So something that is going, 20,000 hertz, for example, or whatever, assuming we can hear it, the auditory neurons, turns out it fails to phase lock. And so reading the actual firing rate in this case is completely useless. And so that's why we wrote, at high frequencies, then we can only retrieve frequency data, as in the physical stimulus frequency, from tonotopy. So yeah, you can just kind of conclude that from, well, it's not any curious reason, but it's specifically due to it's a physical limitation of our neurons that we just can't phase lock when it gets to be super high. Yeah. So that's it, really. There's nothing to it. Okay, Arcelia. Zoe. That's Zoe's name. Could you explain concepts of simple and complex cells? Oh, okay, sure. So we're coming back to vision now. Gosh, we just keep bouncing around to. Oh, where's my? I can't find where's my cursor? Oops. Oops, it's not. It's this. Ho hum. Yeah. So the okay, all right. So the major umbrella difference between simple and complex cells is that one requires the bar not only to be at a specific orientation. Don't ask me, guys, why I'm still holding on to this screwdriver. We require the bar to be a specific orientation, but it also needs to fall in a specific location of the neuron's receptor field in order for me to fire. In a complex cell, I need a specific orientation, but the specific orientation can swipe anywhere in my receptor field for me to fire. That's the like overarching big difference between the two. In terms of construction. So I mean, I actually don't need to give you my poopy drawing, right? It's actually here. So the question is how can we end up with a neuron that has a receptor field that is so selective, both in terms of orientation and in terms of position. And so it turns out that we will be able to like if we were to physiologically test, via single cell recording, I'm poking into a V1 neuron and I'm very, very carefully, I think we showed the video, we very carefully just map regions of, okay, it stopped firing, and then, okay, here we start to be a plus area, and then a minus area. If we carefully map that, we find we'll have configurations like this. So in this particular case, this would be an example of, whatever orientation this is over here. I don't know, 10 degrees, 15 degrees, right? Whereas if we had, a vertical tuned neuron, then the RF will probably look like this. So plus here, and then minus like that. I think it's hard to think about because I didn't really talk a lot about, hey, how does a V1 neuron just magically end up with an RF that is like this, right? We talk about this actually a little bit in my other course, but I don't want to confuse you, but imagine that the LGN cells coming before it are smaller, and they will have also their own donut-shaped center surround configuration. Sometimes it's like this, sometimes it's like this. So this part won't be tested because I didn't teach it, okay? But just FYI to kind of complete your knowledge. So LGN neurons will look something like this. And so the general rule of thumb is, in terms of the receptor field, they get bigger and bigger the further up we go, the cascade. And so it turns out, in order to of course, LGN neurons are the ones feeding to V1 neurons. And in order to kind of construct the shape, it turns out what we have is basically a bunch of center surround LGN cells that are lined up. So if you can imagine, this here is created by a whole bunch of donuts like this. So we've got, like, let's say 5 LGN cells, and each with, the plus center. I don't know if you can even see that. Each with a plus center. And so if you imagine, like, these 5 cells are the ones feeding into V1, that's what gives V1 that vertical responding property. And so similarly, if we wanted to create this, just imagine again a bunch of donuts lining up like this. We've got 5 LGN cells that are, being read by that one particular, 15 degree preferring V1 neuron. And it's because these are the 5 that this particular neuron is reading off of. This is what gives us this 15 degree preference. All right, so Zoe, that's how it comes about. Because I know sometimes, having chat with you, I think it's a little bit like, it's because of the details that kind of make you a little bit confused, right? So for you, all you have to understand is, okay, we've got these off regions and on regions, and depending on where they're located in the receptor field for that particular neuron, that gives it its particular preference. Complex cells are actually a little bit less complex, ironically, because it actually doesn't really have this kind of
on, off, on, off kind of configuration. It actually just is a big, it's a big WADA thing that is, just plus, plus, plus over here, right? So it's going to be orientation selective, but it's anything that happens just to fall in to my RF at that particular orientation is already sufficient to make me fire. So orientation selective, but respond optimal to this particular oriented bar, so long as it occurs wherever in my RF. Is that okay, Zoe? And I think I did also say there are more categories that get a little bit more specific beyond these simple and complex cells. I mentioned it again. It's not in our notes, so I'm not going to test it. But again, FYI. It's related to retinopathy in the sense that the RF itself, the location of the RF itself, corresponds to this concept of retinopathy, right? Like, so when we're talking about this responsive area in the field, that is the concept of retinopathy. So that V1 cell, okay, let's imagine we're looking out into the world, and this is the fixation I'm looking at because, hey, Vision Lab. And what's going to happen is V1 is not going to actually necessarily respond to the whole area, because there are some that are so, like the very extreme parts, actually V1 is going to have very minimal representation for. It doesn't matter. But the point is, we're now, for every kind of spot in this region, there will be some dedicated neuron that will be taking care of stimuli that falls in that chunk of the visual field. So that chunk of the visual field is that receptor field, okay? And so now we can have multiple neurons. We can have multiple V1 neurons with an RF in this patch, but be differentially orientation selective. So I've got neuron A who actually has an RF here, and I have a neuron B who also has an RF here, but neuron A reads off of a bunch of LGN neurons that gives it a 90-degree preference. Maybe neuron B, although I'm responsive here, maybe I'm a 15-degree preferring neuron. Okay, so yeah, the RF part of it is retinopathy. Right, okay, great. Why are you crying? Oh, okay, I guess because you're probably studying, right? Sorry, guys. Oh, next. Is that a laugh, or that's a laugh cry? Next. Antonius, could you explain the mechanisms for olive neuron? Encoding sound waves, ITD. Yeah, that's so funny, you guys. Your questions are like back and forth between these two lectures. Is it because you guys didn't study the last 3 lectures? Is this why? Are we just warming up here? We're warming up? We're gonna be here all day? I gotta fix that button box. Wrong lecture. Right. Superior olive. Where's that incidence detection thing? Oh, here. You know what I have to tell you about the exam, I'm getting to Antonia's question next. Is that, well, the first thing you can be sure of is that at least every module has been represented in the short answer section. The other thing you can be confident about is that the thing that I said I was going to test, I definitely do test. And yeah. And the last thing I kind of wanted to say was, like, I've sort of said this to your other students who've actually come to review their midterms, but like, it doesn't strike me that most of you don't understand the content. It strikes me that most of you do have a good understanding of the content. I think at this point it is just making sure it's expressed like concisely and precisely, so that what's in your head is what is written down, so that the tutors know clearly to give you that tick. And just make sure you're using all lines. I gave you guys an extra line in all the questions. I don't know if that's gonna help. Use the space, count, do you have 7 unique points? And that's a good strategy way. Like at this point, I don't even think it's about knowledge. I think it's just test taking at this point. Okay, superior olive. So ITD. Now ITD, of course, was about the difference in the arrival time of the source, right? So I think like physically understand where it's coming from, right? So if I have a sound source that is located to my right and this is propagating towards my head, it's going to arrive at my right ear before my left ear. And so the ITD itself is the delta in arrival time between the right and the left ears. So unfortunately, though, something in the brain needs to do that math of the delta. And this math it's not necessarily the case that we do, okay, right ear response, left ear response, and then let's subtract it. We don't do that. For many reasons, that's not efficient. But instead, what we have are these coincidence detectors that fire with arrival time. I think the best way to think about these coincidence detectors they're actually sort of like ITD tuning, if you will. So if you take a patch of superior olive, anything located in the center in this kind of schematic, although it's not really this perfectly organized in real life, but anything located kind of roughly at this position will be firing when there's about a 0 ITD, right? Because we've got a situation where the left arrival and the right arrival will kind of end up, the propagation will have it meet sort of at neuron 2, right? Whereas if we have neuron 3 firing, then this is an indication to your brain that this one was actually propagating a lot sooner, so from the left side than the right side data, right? So you see so this one's coming in first because the sound source, I guess we can conclude that it came from the left side because it's propagating a lot longer, and then sound source looks like it finally arrived to the right, and so this is why this travel time is a lot shorter. Okay, it meets up at this position and causes neuron 3 over here to fire. So in some ways, right, I think a good way to think about it is a little bit like of an ITD orderly representation across the spatial extent of the superior olive, right? So in the center is about equivalent ITD, which leads you to be able to conclude, your brain to conclude that it was either a sound source in front or a sound source behind. If you find that neuron 3 is firing, that leads your brain to also conclude that it must be a sound source towards the left because left propagated first, and vice versa if it was a activity on the number 1 side. So there's kind of this orderly spatial representation that represents this point of coincidence, and this point of coincidence, of course, will change on the spectrum of on the azimuth. Is that good, Antonius? Yeah. So don't think about it as really actively computing a delta. I think that would have been the easiest way if we evolved to do it like that. I think there's a lot of problems with that, obviously. But really it's a spatial representation of the ITD. It's more of that. Okay, perfect. Awesome. Holy cow, 73 WhatsApps in the past 20 minutes. That's frightening. Is there still a possibility? Oh, are you Jack? You're Jack. The answer is no. Yeah, because I tried to offer it, and the department said no because I have to teach 2051. So I said no. I said no to 2051. It's not that I don't like to teach it. I just like to teach advanced perception more. And they said you can't. So So it wasn't an option to not teach the neuro classes. So the class that had to be sacrificed was 3061. You know what? It might not come back actually in any time in the near future. It's unfortunate because actually I resurrected that course like 3 years ago. Antonius, for the modal hierarchy part, is VLO and VLC both referring to the ventrolateral nucleus? Yes, for your purpose, yes. Because there are different subchunks to the ventrolateral nucleus, but I think for our purpose, just it's the ventrolateral nucleus. No need to differentiate for our exam. Could you explain how protein kinases and protein synthesis works in memory consolidation? All right. Hey, someone who actually reviewed the last lecture, which I thought was a lot of fun. That's gonna be by the end somewhere. Is this the right one? Hold on, let's see. Kirk, is this the slide, or it's not really the slide, it's the one after that? Okay, while Kirk is replying to me, I think the overarching story here up to this point was that, okay, we know that what happens during LTD and LTP is that there's this story about AMPA insertion and AMPA removal at the postsynaptic neuron. All right, I already started my story, and and then we came to talk about how. So in this particular slide, it was about, okay, we've actually got two proteins here, one doing the job of the phosphorylating, and so making the existing AMPAs better, and then also driving the insertion of new AMPAs. And then there was the counterpart of that, that's the phosphatase doing the bad stuff, right? So we're making it less efficient, and then we're also driving the removal of. So then Kirk's question is, I guess this stuff, like this stuff here. Right. Well, we can just do this again. It doesn't really matter. Last 3 slides or whatever. So the added twist to it, though, is unfortunately, proteins are not permanent in the nervous system, right? And so we have a situation where, sure, it's good that LTP happened according to Hebbian principles, and I've strengthened the synapse, but then unfortunately 2 weeks later, the thing that I've strengthened, like the substrate that I've strengthened, have just died off, right? Because it's got this 2-week lifespan. So here at the very end of that lecture, I was trying to go over some mechanisms to kind of help preserve memories and help consolidate memories given this kind of 2-week lifespan limitation. So the first prerequisite really, in fact, I think should have been, I should have reordered this. I should have talked about like number 2 first, because certainly, the first job is that we've got to synthesize more, whether it's going to be AMPAs or kinases, whatever, there's got to be some directed synthesis to make more of it after it dies off in the 2 weeks, right? And so I think this is sort of more about this slide at number 2 here. So we've got to make more, and then we'll come back to number 1 where we keep phosphorylating. And to make more in the nucleus, you can imagine that we've got actually certain genes. This is one example, specifically this CREB, which is actually what it stands for here. I've just written out for the cyclic AMP RE. And CREB, you can imagine, really has 2 configurations. We've got the cyclic response element, CRE, over here, that can be sort of capped off by this CREB2 element. And in this configuration, no transcription will happen. So what happens when we're directing protein synthesis to keep replacing, let's say, the AMPAs that have gone off and died just due to natural recycling, will direct CREB2 to be replaced with another configuration, this kind of CREB1 cap instead. And so when this CRE, this cyclic response element, is capped with CREB1 rather than CREB2, then this is a signal for your nucleus to go and transcribe the particular protein. So this is just an example way of signaling, please produce, to your nucleus here. That was the deal with the CREB. And so then, because that was in my red line notes there. And so there, after I have synthesized, the deal is, though, that at this point, I'm sort of like a neutral AMPA, right? Even if you've inserted this new AMPA into the postsynaptic membrane. So at this point, though, we need the same cleft to remember to phosphorylate it back to kind of strengthen its efficiency, right? Because this is my originally strengthened LTP, strengthened synapse. I've had some AMPAs that have died off due to life cycle. I've now directed CRE to go and do its thing and generate new AMPAs. It's coming and inserted back, transported back to my postsynaptic membrane. But now I need to rephosphorylate, right? And so this slide here actually is a couple ways in which we can keep phosphorylation an ongoing thing, right? So it's not just suddenly detect, say, CA influx through an MDA, and then we're going to one-off shot the phosphorylation and then it's done, right? Because that's going to be totally useless. We need a continuous phosphorylating situation in that particular important synapse. And so these are two example mechanisms of how we might keep phosphorylation on, right? Number one is the CAMK2, which is the one I was kind of quickly talking about here, right? CAMK2 is an example of an auto phosphorylating kinase, right? These are both kinases here. And CAMK2 is, you can kind of think as this kind of jackknife configuration. Regularly, when it's not phosphorylating, it'll be like this, kind of in its closed shape. And when it is active, then it will kind of open up like this, and you can see how there's this kind of catalytic region where it will be open to phosphorylating whatever AMPAs. And what happens with the CAMK2s specifically is that it also has this capacity to keep itself open, so that it will kind of, you can see this kind of phosphate group that has been attached to this open jackknife. And in this configuration, the point is that CAMK2 can phosphorylate itself, resulting in a perpetually open CAMK2. So it'll just sit in the cleft, and because it's autophosphorylated, it will keep being available to phosphorylate new AMPAs that get replaced. All right. Is that good, Kirk? Cool. Samuel, we talked about you. Yes, both BLs. For your purpose, just understand both to be ventrolateral nucleus. That's the main nucleus in the main structure in the thalamus. Main structure in the thalamus for the motor system. Ventrolateral nucleus. Yeah, so I was just saying earlier, because it is specifically referring to different subchunks of it, but for our purpose, let's just keep it simple. They're both talking about the same broad structure anyway. Both ventrolateral nucleus. Okay. Anything else? Which is the hardest module in the second half? Which is the one you're most worried about? Maybe second last one. Everyone. the visual system. Oh God, I wrote the thing yesterday, but I can't remember if Yep. Well, yeah, I told you there's going to be at least one short answer on each module. So there is definitely one on the visual system. Anything AMPA related. Yeah, I don't know how to do this without giving any hints. Like typically when I test, I spent a considerable amount of time on in class. So generally, like a very small detail I wouldn't really be too worried about. Yeah, I think conventionally the last two you have said that we all know that you have said. Okay, let's give you this. I wonder if it's because of the, like, in the visual system, is it because of the, like, different tracks and the different nerves, and then there's that, like, is it like the is it the, like, the visual field representation that's like it's so confusing? Is that the point? Because if it is, I would advise you to so I can tell you that as I remember, there will be an MCQ that might tap into that, but like the pathway stuff, but there won't be any short answer. Like, I won't ask you to reproduce that. As I recall, there is one MCQ that asks, like, if I lesion here, what do you expect will happen? But that doesn't come back up again. I spent a lot of time with everything. I mean, like, for example, the slide we're looking at here, right? I don't know, I spent a few minutes on. Like, I wouldn't really go, can you give me the 2 protein kinases that we talked about. I think I'm gonna start to give away too much soon. I also have a very bad memory, so what I wrote yesterday I don't entirely remember already. You can kind of try to prod me. It might not work because I don't totally remember. The bonus question is worth up to 2 marks. I don't know why some people didn't even try. It's not like we subtract marks for incorrect answers. So there's a bonus. It's worth up to 2. Holy sh... Could you print those diagrams in question? Oh, in the questions, if there are diagrams, can we do it in color? Yeah, okay, so that's easy. There are no diagrams in the final, or there are no pictures either. Oh, you can draw your conclusions as they may. I've also reminded you guys, like, even though, what are the bonus? That's nice try, Zoe. That's a very nice try. Why would I tell you? Like, then why are we doing this exercise? I can tell you that this bonus question doesn't hinge upon, like, one word that I said, or anything like it. It's conventionally, that's usually how I did my bonus questions. Like I kind of try to catch, like there must be a week where I said something really stupid and then we catch who's in class. This time it's not. This time it's not. This time, like I highly encourage you to at least give yourself 2 minutes on it. Not like 10 minutes, like 2 minutes. Maybe 1 minute. Just like go have a look before you hand it in, okay? Dear God. Keep trying, Zoe. Keep trying. Oh, is that how you were trying to predict my bonus? Yeah, you know what? So, like 1 year, I was, like 3 years ago, I think, I was discussing my pure disdain for Deliveroo, and like, I don't know what happened. It was this big, long story, and it actually appeared on the final. It appeared on the final. So I gotta tell you guys, you know, when we send sample exams for external external review, the admin staff refuses to print my last page because it has the bonus question. Because they say they don't know what the reaction of the external will be. I was like, why do we care? Like it's not that easy, Samuel, but it's like it's pretty easy. Like you need to show some minimal effort and there should be something. And so it's a question where it's scalable too. Like it's 0 to 2. For you'll see. And like, you know, the reason why it matters, guys, like 2% is worth like 4%, you know, of your final total. I don't know why no one cares. Oh no 2% is worth 4% of your exam grade. Yeah, out of 100, right? So hold on, let me just, right, it's worth like 2 marks out of, is it? Yeah, it's worth like 4% of your final total in the exam. Like I don't know why you would not read it. I'm so upset. Like you spend all this time making the bonus question, you're like, the meme pick on it. I thought about it. You know what? Actually, I did think about it. But I'm not. No. Wouldn't that be sad though if someone got it wrong? I don't know. Maybe you guys save paper and just don't print out that slide every week. 1 year, Larry was a part of the bonus also. And yeah, people got it wrong. But when people get it wrong, that's when you start identifying names who have never turned up to class. Every year there's a few. Oh, I've always thought about what is your lecturer's name. Like I've thought about that because I thought it would be compelling to find out if you don't even know that much. What is 8? Oh, how many Larrys have been there since 2001? Yeah, I probably missed one. So, like Justin took Larry and Larry understudy, and he said, "I'll keep them alive till next year." I don't really hold up any hope. I don't think it's gonna happen. What time is it? Any more academic questions? Or non-academic questions? Larry's pretty academic. Do we have neuro students who are stuck with me next semester? I can tell you about that course a little bit. What is the color of the glasses the cat is wearing on the first slide? I don't even know the answer to that question. What is the color of the glasses? Holy cow, it's black. Zoe, it's me and you again. It's gonna be a small class because this is the first year, and new intakes from this year are the only ones that must take it. New neuro intakes. But I can tell you, Zoe. Yeah, it's me. I teach all courses in this program. Why are you asking? I teach everything on your everything in your program requirements. You know what? Actually, I think you guys might like it better. So there's some we're wrapping up kind of the foundations, like knowledge. So we're gonna do a little bit of homeostasis because we haven't really talked about stuff that we don't really voluntarily control, right? But certainly your nervous system plays a huge role in that. So we're going to touch base a little bit on that. So a little bit homeostasis and endocrine control. But in fact, it's looking like we'll just have like the one quiz on like content, and we're going to spend a good chunk, like maybe 4 weeks, teaching you how to we're doing what I call skill development for neuroscience research. So we're going to learn a little bit of programming skills, actually, and there's going to be a really cute exercise. I'm still working it out. Actually, if you've been to my office, you might have seen hints of it. There might be remnants of some parts. That's because I was building it and I was testing it before I gave it to you guys as a project. It'll be fun. Like, I know, like, when you hear about programming, it kind of sucks. It's not really like that hardcore. I just want to introduce you to the possibilities. We're gonna have some assignments, and we're just gonna do fun stuff with it. And we're gonna see what you can create and what monsters you end up creating by accident. So it'll be a little bit of it's some skill development, and then but we will start off with some, like, just some lecturing. I'll be teaching a little bit, not a little bit. I'll be teaching you all of it. Oh yeah, so the gummy bear is like a prominent feature here. So I did refill it yesterday, but like this is basically what I eat every day. So yeah, and the hand sanitizers. Yeah, you were supposed to sanitize your hands before, but no, never mind. It's just decoration now. Yeah, so I'm like 90% gelatin, but most people know that already. Yeah, okay, so yeah, it'll be like, it's a super small class. I think maybe we're gonna, like, they're gonna be like 10 of us, maybe, not even, I don't know. So I did think about, like, do we actually really need like such a formal setting? But again, I think we also have too many people, so it's not like we can just have a meeting in my office every day. So we'll have a classroom and stuff, but it'll be a pretty close-knit type of deal. But yeah, it won't be totally like O1. It'll be like, half like O1 then and half skill development. I did kind of finish the syllabus. I just haven't really done anything about it. Do we have anything else? Still studying, aren't you guys? Anything else you want to try to pry from me about the final? I reserve the right to ignore you. I'm also teaching perception next year, so as we all know, that's the reason I can't teach advanced perception, because my quota is full. So I might see some of you guys there. That's gonna be a full class. Is it hard? If you took 2101, the perception is gonna be easier. Because perception's not geared for neuroscience students. Perception's a part of the psychology program, right? So there'll be, like, we do several modules on the visual system, but it's not like several modules on the neuroscience of the visual system. There's going to be a little bit of like the physiology of the visual system, but then a lot of it is about more of the higher order stuff that the psychology students like, right? So we're gonna visit like the color system in detail. I think we're doing like depth, motion, stuff like that. But then we're also going to do the auditory system in more like higher level stuff. This was cancer. What are you talking about? What are you talking about? You go find me a better neuroscience course. Ow, that hurt. Ow. I mean, if you wanted to learn like fake neuroscience, you could have taken biosci, right? or maybe you have taken biosci. You didn't have to register for this course. I'm not bruh. I'm sister. Yeah, Arcelia, I think you'll like it. It's much less hardcore, just because the audience is different. The audience is not neuro. Yeah, well, that's it, right? Because you have to think about the audience. Biosci is for psychology students, right? Foundations 1 is for neuroscience students. It's way too easy. Who teaches biosci? Should I tell he or she that? But you also have to think about from a program level, right? We can't really run a neuroscience program and teach you biosci, and then you go register for a graduate neuroscience program. You're like, oh man, I'm at the bottom of my class. I'm not ready. I'm not ready. You're not going to be competitive, right? So there's a reason why, if you register in a neuroscience program, we're going to teach you neuroscience. Come on. Get over it. Okay. All right. Well, if you have nothing you really want to try to pry from me, gonna call biopsych fake neurosci for now. Oh my God, Yasmin. That's gonna piss off so many people. But that's what it is. Let's call it as it is, isn't it? How many courses do I teach? 3 every year, except for last year. I taught 2. That's why if somebody took perception last year, it was not taught by me. So yeah, 3 a year. So I do 01 in term 1, and then 03 and perception next semester. And then I supervise like 1 million students that I don't remember the names for. It's really bad. You can ask KY. Every single, the start of every lab meeting, it's just sort of a reintroduction. Tell me who you are again? Your intern or your thesis student? It's not that I don't care. It's just I'm really bad with names and faces. Though I'll give you a history lesson. I used to teach 5 my very first year at Hong Kong U. That was KY's last undergrad year. That's how old she is. So now you guys can calculate. I can tell you I started 5 years ago. Okay, is that her age now? That year I taught 5. Your message was held for review. Ta-da! That's right. That was her last undergraduate year. My very first year. I should try to facilitate the knowledge of memory consolidation. I do. That's what I do every week. But you guys need to be awake for that. Though, wouldn't it be really funny if we really could, one day, there's just a way to, everybody, let's get in line, everyone get a little bit of TMS. And then it's that'll be fine. It's going to last 48 hours. You'll be all right. I'm just imagining it doesn't work right now. Cool. All right. Well, then, we'll close it in 5 minutes then. Come on now. Fire those last questions at me. As you can see, the room is in a bigger disaster than it was last week. This is really bad news. I actually want a weekend this weekend. Yeah, I used to teach 5 courses, and I got all the stats courses too, and then there's perception, and then stats. I taught all of 104, 2060, you name it. I've probably taught it in this department. This is what they do to new people, right? New people get all the courses that nobody likes. I've said too much. We're oh my God, we need to take the video down too. Come on, what else do you want to know?
Got a few minutes. You can ask KY's in the exam. You can ask her if she thinks it's hard. KY, do you think the exam's hard? There's no reply. A little bit. A little bit. I think the MCQs are even easier, I think. The short answer, I don't know. Like, I don't know how to say that it is harder. It's just like, I mean, what I promised to test, I do test. There we go. Guys, I've said how many times that LTP, LTD poop in the AMPAs and the NMDA, that will be one of them. So you might as well just pre-generate a 7-point answer for that. You might as well. Bonus question is... It is not. It's probably the easiest bonus I've issued in like 5 years. I honestly can't even remember what the visual system one is, but like I don't think it's that bad. Oh, I kind of remember it now. And then there's obviously one on the motor system. Noted with thanks. That's KY's fault. It's not. It's like about the same. It's about the same. Minus the diagrams. No diagrams. I don't know how to say anything about the exam without giving you all the questions. Oh, I mean, like, if you want to see the state of my room, like, like, back here's not looking hot. I've got a whole set of, like, screwdriver things, like, rocking over here. I'll, like, spin around my webcam. Like, it doesn't look hot right now. So there's, like, hold on, what can you see right now? What can you see? So there's, like, oh, God, this is, like, so delayed. Let me just see this. So, like, you're looking at the plant that dies every 3 days. And then you can ignore everything else. That's right, you do see a minion over there. But he's wearing 3D glasses. But, like, over here is, like, a whole bunch of, like, this is a new portable eye tracker we bought. I haven't really set that up. Over there is, like, okay, so there's a reason why that shelf is by the door. It's because, like, important people can't notice all those toys there. So when they open the door, they can't see it. And then, like, in that gray cabinet there, in the first, it's not even locked. It just looks like it's locked. And the first thing, it's like, it's all snacks. It's basically everything unhealthy is in there. It's a situation in the office. Situation in the office. So I was gonna, actually, I have a bunch of things to do before I can even clean. I've got to submit a paper. That's why you will work harder. That's right, Jack. Study. Although we said it's too late now, right? Like, we're now trying to consolidate in 48 hours, 72 hours. Okay, so as you are finishing studying this weekend, if there's something that comes up, email me. You can trust that probably I will stop replying to emails by like midnight of Monday. So that's too late, okay? Everybody, let's just, we're at peace. If, like, you're still studying after midnight on Monday, then it's just, we're at peace with the fact that that's just gonna be the way it is. Okay? Okay, well, try to have a good weekend. I know probably you're studying for more than this course, but so just ping me an email. I'll be here, not cleaning. Maybe not exactly here, but all right. Bye, guys. Bye-bye. Bye, Zoe. No problem.